Okay, good morning. I'm Cyrille from OSIAC in Cameroon, and I'm happy to share with you the data we got from the study investigating Anopheles pneumoniae susceptibility and immune response to Plasmodium falciparum infection. So Anopheles pneumoniae is an important malaria vector in sub-Saharan Africa. We got up to 11% infection rate in this mosquito. The mosquito is high endophilic, highly anthropophilic, and in some area, this mosquito is responsible for up to 80% of malaria transmission. The mosquito is also widespread across Africa, and you can see in red. And it brings in large permanent water with emergent vegetation. But this mosquito is less studied because it is difficult to colonize in insectary, but progress is being made, and we use now the forced egg laying method to allow to work on failed mosquitoes, notably on insecticide resistance, using the method of Morgan and collaborators described in 2010. Also, genomic tools have been described in these species, and we can mention macroarray, genome-wide macroarrays, and such progress allow to elucidate molecular basis of key phenotype in this mosquito, such as insecticide resistance. But to date, little is known about the Anopheles pneumoniae immune response against Plasmodium falciparum. So the only two studies carried out to date have used the murine parasite and the mice feeding system. And in our studies, we aim to assess the infectivity of Anopheles pneumoniae natural population to Plasmodium falciparum infection, and to investigate the molecular basis of Anopheles pneumoniae immune response to Plasmodium falciparum infection. I would like to mention that Plasmodium falciparum is the natural malaria parasite, so it will be more realistic to use this model rather than the laboratory model using the murine parasite. So what we did is that we collected mosquitoes in Cameroon in a site situated in humid savanna area, and we use electric aspirators to collect mosquitoes inside houses, and after collection, we transported this mosquito to insectary over 350 kilometers in cooling box. And once in insectary, we allow gravid female to oviposit individually using the forced egg laying method. And after that, we proceed to morphological identification, and we confirm that with the molecular identification. And after that, we pull Anopheles pneumoniae progenies, and we rear them until adult. We try to assess the infection of mosquitoes by using ELISA techniques. And for infection experiment, we first identify a gametocyte carrier in children in an area with high transmission, and we examine 1,017 children between 4 and 11 years. So the blood sample was screened by direct microscopy, visualization of Plasmodium parasite on tick blood smears. So we quantified trophozoite density using the semi-quantitative count method, and gametocyte density was estimated by microscopic read, again, 500 leukocytes, assuming a standard white blood cell concentration of 8,000 per microliter. After that, we collected 400 microliter of infected or non-infected blood meal into a parenteral tubes. We centrifuge this blood meal at 37 degrees and we replace the serum by the non-human one. And then we feed mosquitoes using glass feed system with paraffin membrane at 37 degrees during 30 to 45 minutes. And we use the non-Plasmodium susceptible Anopheles colusi to monitor our blood handling procedure and the infectivity of our gametocyte. So after blood feeding, we remove non-partially fed mosquitoes and we kept mosquitoes until day 7. We dissected mosquitoes and we tried to observe the oocysts in milk gut using microscopy. And we recorded 5 parameters: the feeding rate, mortality to day 7, infection prevalence, and oocyst load in milk gut. For transcriptomic analysis to detect gene associated with Plasmodium infection in Anopheles funestus, we use this scheme to make comparison. So what we did is we use the 8 by 15 KB microarray chips, and we analyzed the data using GenSpring software. So as first result, we collected around 4,400 mosquitoes, and we got high survival rate to insecticide and high oviposition rate, and we also get high hatch rates. And this means that almost all mosquitoes were already inseminated at the time they were collected. According to morphological identification, followed by molecular identification, we observed that we got 3 species in our area, namely Anopheles funestus, Anopheles gambier, and Anopheles faroensis. But Anopheles funestus was by far the most abundant species collected. Looking at the natural Plasmodium infection of our population, we analyze mosquitoes by ELISA, and we observe high infection prevalence in both vectors, Anopheles gambier and Anopheles funestus. And this showed that there is high level of malaria transmission in this site despite high coverage of long-lasting insecticide net, which is up to 80%. Regarding Plasmodium infection prevalence in children, as I said, we examined 1,070 children between 4 and 11, and we observed high prevalence of infection in these children, around 50%, but the gametocyte carriage was low. So concerning feeding rate, we observed that blood feeding rate is while F1 Anopheles funestus mosquitoes was low, and this is similar with other studies carried out with wild F1 mosquitoes of other Anopheles species. And however, what we did is that we compensated this by increasing the number of mosquitoes in infection experiment. Concerning the infection rate we got, we can see that 11 out of 14 experiments, we got at least 1 infected mosquitoes. And you can see that we obtained in some, in 1 experiment, we obtained 100% of infection. And also the oocyst rate was high, with the mean of 7 oocysts per megagot per megagot. So this shows that high infectivity of our gametocyte and high susceptibility of the wild F1 Anopheles phoenicus mosquito to Plasmodium falciparum infection. We try to see if there is correlation between gametocyte density in blood, mosquito infection rate, and oocyst load and mortality. And what we observe is that there was a general trend showing that infection rate and oocyst load increased with gametocyte density in blood sample, but this correlation was not statistically significant. When we compare the susceptibility of Anopheles phoenicus to that of Anopheles colusi, we show that the feeding rate in Anopheles colusi was high compared to Anopheles phoenicus. And this is normal because this Anopheles colusi strain is now well adapted to feed in insectories. It has been maintained for over 10 years in insectory. But when we compare infection rate and mean oocyst load, we didn't observe significant difference between both species. So comparable level of susceptibility to infection observed in both species reinforce the view that they are readily infected with Plasmodium falciparum, the most common and dangerous species in sub-Saharan Africa. Concerning transcriptomic analysis of Anopheles phoenicus immune response to Plasmodium infection, here you have a Venn diagram comparing with the 4 comparison. And what we can observe is that when we compare infected mosquitoes and non-infected mosquitoes at between 24 hours and 7 days, we have more than 1,500 genes differentially expressed between samples. But when we compare infected samples and non-infected samples at day 7, we just got 30 genes differentially expressed. And this means that the immune response is not maximal at day 7, but this seems to be the case at 24 hours post-infection, because we got 222 genes differentially expressed. So we know that at 24 hours post-infection, this is the time when the okinet crossed the midgut, and many studies show that this is the time where the immune response against the parasite is maximal. So we try to focus on that time point. And what we show is that comparing infected and uninfected mosquitoes at 24 hours, we got 144 genes upregulated and 78 genes downregulated. But at day 7, we got 8 genes upregulated and 22 downregulated. And we can see that only 4 genes are common between the 2 time points. And this confirms that the immune response against Plasmodium infection is maximal at 24 hours after injection of the gametocyte, the time when the okinet crossed the midgut. So looking at the gene differentially expressed, what we saw is that we observed 5 non-immune genes in Anopheles gambiae, also found differentially expressed in Anopheles phoenicus infected mosquitoes, and this gene was upregulated. And we also observed other putative insect immune genes differentially expressed in Anopheles phoenicus infected mosquitoes. However, TEP1, a major immune gene in Anopheles gambiae, was not found differentially expressed in Anopheles phoenicus infected mosquitoes. So to sum up our study, we can say that the present study indicated high malaria transmission in our study site, despite light coverage of long-lasting insecticide-treated net. High and similar level of susceptibility to Plasmodium infection was observed in Anopheles phoenicus and Anopheles colusi in parallel experiment, and this reinforced the view that both species are equivalent in transmitting the malaria parasite in sub-Saharan Africa. Preliminary macroarray result show that APL-A, APL-CL1, and CTL4 are probably involved in immune response to Plasmodium infection in Anopheles fenestus, similarly to what has been reported in Anopheles gambiae. The fact that TET1, the major immune gene in Anopheles gambiae, was not found differentially expressed in fenestus-infected mosquitoes suggests that some difference may exist in immune response in both vector to Plasmodium infection as observed for insecticide resistance. We know that in insecticide resistance there is KDA mutation in Anopheles gambiae, but this was never found in Anopheles fenestus mosquitoes. So the next step is to do some qPCR and sequence analysis. And to finish, I would like to thank all my supervisor, my PhD student, and my team, and thank also the Wellcome Trust who funded this study. Thank you.
Any questions for Cyrille?
No, we don't know. We don't know anything about Plasmodium vivax. So this is just the first study on between fenestus and Plasmodium falciparum, but nothing is known about vivax.
Sorry, Albert.
What is the level of homology of the TEP1 from Anopheles fenestus to TEP1 of Anopheles gambiae? I was wondering, maybe they are pretty different, and was there another TEP that was differentially expressed in fenestus?
Okay, thanks. This is what we are trying to find out now, to see the homology, but also we plan to do some sequence analysis because sometimes the immune response could not be due to the level of expression, but also by some mutation which can change the gene, the function of this gene. So we are trying to find it out. Is.
Beautiful work, Cyrille. Thank you. Have you any information about the blood meal of mosquitoes collected in villages in which you have found this high sporozoite rates?
Yeah, what I didn't mention is that we tried also to assess the origin of blood meal, and what we were able to identify at 80% of blood meal, and all were taken on human. So
80%?
yeah, yeah, on human.
How do you explain this with a high coverage of the nets?
Maybe the net are not properly used, because if you see some picture, you can see that people are using net for fishing, and sometimes as I don't know. So sometimes net are not properly used, but also we have some hole in net, and also this could be because of high insecticide resistance in this site, because we saw that there is increasing insecticide resistance in this site.
Thank you, Cyrille.
Okay, thanks.